Welcome back from this virtual coffee break, and I suggest to start immediately with the first speaker of this last section of today, who is Gerhard Thiel, who is hi Gerhard.
Hello.
Hello. Who is a professor at the Technische Universität of Darmstadt. He will tell us about the physicochemical basis of channel gating in potassium channels. So please, yeah.
First of all, thank you for the invitation. I hope technically everything works and you can hear me, you can see the slides. I'd like to sort of link to some talks of today and yesterday, and I want to present our view on how potassium channels may work. The work has been done in my lab, and there are three main people, Oliver Rau, Intra Schröder, and Peter Hansen, who contributed to this. All of this has been done in collaboration with Anna Moroni's lab in Milano. The computational work was done at the University of Dortmund with Stefan Kast. And we have a long-standing collaboration with a virologist, and you will see later why this might be important or why this is important. We are coming from the experimental side. We measure ion channels. We identify in the 12 time histograms different open and closed states. Here, an example with 3 distinct closed states, a long-lasting, long-lived closed states, a medium, long-lived closed states, and very fast closed states. So from this, we usually generate models with an open state, with 3 closed states, and with rate constancies, which are leading and going into or out of this open state. Now, in the era of structures, everybody wants to relate these states, the open and closed states, and also the rate constancies to conformational changes in the proteins. And here, the gold standard for potassium channels, the KCSA channel, the first real structure of a potassium channel. We were by serendipity very happy and very lucky to find about 20 years ago a channel which is even much smaller than the KCSA channel. The monomer of this channel only has about 82 amino acids, and this is enough to do all the work of the pore of a potassium channel. So we have a minimal version of a potassium channel, which is a good model system for the function of a potassium channel, because it has inherent all the functional and the structural entities of a potassium channel. So this channel comes from a virus, and you see just on the alignment of the pore that it is very, very similar to the KCSA channel, 2 transmembrane domains, a pore helix, a selectivity filter, so a lot of similarities to the KCSA channel. So we think that this is the ideal system to really understand the basic ABC of structure-function relations in a potassium channel, because it did not go through any co-evolutionary developments, so it's free of the need of the host. It's really sort of a minimal version of what the pore of a potassium channel can do. And we are sort of lucky in the sense that the channel is very small, which means the degree of freedom of interpreting structure-function relations is very limited, because there are only 82 amino acids. And the other advantage is, because it's a viral channel and viruses undergo a lot of mutations, we have a big library of different variants of this channel. So we have more than 100 different amino acid sequences of this channel. So now we can measure this channel, and the system is again very simple and very reduced. We use in vitro translation, and we do the protein synthesis in an Eppendorf cup. we synthesize the proteins into these small nanodiscs. These nanodiscs can be isolated. They contain a his tag. And this unit of a nanodisc, which contains one single channel, is then incorporated in a cleaner lipid bilayer with the result that a single channel inserts in the membrane, and we can study very nicely this single channel. And now I show you the power of the system. You see here two different amino acid sequences of two different channels. And you can see that they are quasi-identical. There are a few amino acid exchanges which are either very conservative or semi-conservative exchanges. So from the, let's say from the amino acid side, you would not spend a lot of money of doing mutations. But when you compare these channels, you see that they operate in a different manner. One channel has a very high open probability. The second channel has a very low open probability. When we analyze the data, we see that the single channel conductance, the unitary conductance of both channels is the same, but the open probability is very much different. You see that the one channel has an open probability around 40%, while the other channel is most of the time open. If we now look at the 12-time histograms, we see the reason for this, because this channel with the low open probability exhibits very long close states. And these long close states, they last, oops, what happened now? These long close states, they last up to several hundred of milliseconds, while this channel with the high open probability is missing this long 12 times. So we have now two channels which are very similar, and one has a long close state and the other one is missing this long close state. The question now is, what is the reason behind it? Now we can go and analyze the differences, and we see that most of these amino acid differences are in the transmembrane domains. So we swapped the transmembrane domains, and we realized that the difference in open probability goes together with the structure of the second transmembrane domain. And after doing a lot of mutations, we found that the presence and absence of this glycine and this serine is the key for the open probability. We see that the presence of a serine is reducing the open probability, and we see that the presence of a glycine is enhancing, is opening the channel. So the question is, why does a glycine open and a serine close the channel? And the first thing which comes to mind is some general information for from ion channels, from potassium channels, in which people have identified a so-called gating hinge, which is mediated by a glycine in the inner transmembrane domain of these channels. And it has been proposed that this glycine is instrumental for the closing and the opening of this inner part of the potassium channel. So the first idea was, we identified also in the small viral channel a glycine as a gating hinge. The experiments did not prove this, because when we exchanged the glycine for another amino acid, we did not lose the high open probability. So glycine alone cannot be the solution, cannot be the answer to the question why the open probability in this channel is so high and this one is lower. We then went and we made many more mutations, and we saw that when a serine was here, the open probability, when with no serine, the open probability was high, and as long there was a serine or a threonine, the open probability was low. So it's not the glycine, it's the serine which is important in this position for determining the open probability. And the reason becomes clear when we look at some structural papers which show that amino acids cannot only make interactions with neighboring units, but they can also do intrahelical hydrogen bonds. In the case that a alpha helix contains a serine or a threonine in a gauche position, you can find that you can find hydrogen bonds between the serine and the upstream amino acid, the backbone of the upstream amino acid, which is either 3 or 4 amino acids upstream. And the result of this intrahelical hydrogen bond is that the alpha helix goes from a straight into a kinked position. So serines and threonines, we learn from this paper, they have an impact on the general structure of alpha helices, and this could be an explanation for a gate. But by itself, there's probably not enough to have a kink and to produce from this kink a gate. So the idea is maybe in this alpha helix, the serine is interacting via this intrahelical hydrogen bond with the isoleucine 4 amino acids upstream to make this hydrogen bond and maybe to make a kink, and that the presence or absence of this is determining the open probability. Again, the question is, how does such a kink mediate a gate? And the answer comes from the MD simulation, because we wanted to see if this is really happening. And in this case, we already had a homology model of the channel, and we didn't even have to do the MD simulations because they were already done. We only had to go and search in the existing simulations if what the data predict is really happening, namely that you have a hydrogen formation, bond formation in the inner helix of these potassium channels. And the simulations were confirming our suspicion. You see here the making and the breaking of hydrogen bonds between the serine and the isoleucine in the inner transformer domain of this potassium channel. So we see that sort of helical bending, which is mediated by serine and threonine, can be sort of instrumental for the formation of a gate. The question is now, how does this close the channel? And the answer comes when we look at the channel structure, and then we see that in both cases we have a phenylalanine just downstream of these critical amino acids. And when we look down in the structure, we see that this phenylalanine could be obstructing the ionic pathway. So the idea was maybe the phenylalanine is out of the way if there's a glycine, and it's in the way if there's a serine. So we tested this hypothesis by mutating the phenylalanine into an alanine in various conditions, either in the presence or in the absence of the glycine. And we found that whatever we did, whenever we mutated this phenylalanine, the gate was open. Yeah. So we further tested this in our MD simulations by looking at, yeah, Anna, I'm talking. Sorry for this interruption. I think there was a probably technical problem. So we looked at the distance and the angle of these aromatic side chains of the phenylalanine and looked at the position in the protein as a function of the presence and the absence of the serine. And these are the results. So we find that in the case that the channel has a low open mobility, the opposite phenylalanines, they move closer together if there is a serine. With the glycine, they are sort of have a much bigger space to move, while the presence of the serines brings them closer together. This is true for the opposite, but it's also true for the neighboring phenylalanines. So the explanation for what we have here is that whenever the serine makes an intrahelical hydrogen bond, it creates a kink in this inner transformer domain, and this kink is moving a phenylalanine in the obstruction or as an obstruction into the ionic pathway, and this probably is making a p-stacking of these phenylalanine rings, and is in this way obstructing the ionic pathway. So in this way, we think that we have solved, or we have identified, a new gating mechanism which is based on very, very simple chemistry. The model predicts that if this happens, there should be really a measurable kink in the alpha helix. We could not measure this kink, but we could experimentally test it. Our assumption was, if this inner membrane helix here is kinked because the serine creates this inner helical bond, then maybe if we put cysteines at the end of this protein here, the kink will prevent the formation of sulfate bridges here. And this is exactly what happens. The channel which doesn't have a serine doesn't care when we mutate the last amino acid into a cysteine. It doesn't care about the presence or absence of DDT. It always has a high open probability. The channel which has a serine in this position, which is promoting the kinking of this inner transmembrane domain, is able, or in this case, in the absence of DT, probably forms a sulfur bridge here and closes the channel. And when we remove this bridge here by adding DTT, it opens the channel again, and it gains again a high open probability. So we think that by really looking at two channels, which at first glance do not look very different, the amino acid sequences are very similar, we can identify a new gating mechanism, which is comprised of a door. The door is a phenylalanine, which by itself is not functional as a door. It only functions together with a hinge, and the hinge is, in this case, the serine, which kinks the alpha helix and moves this phenylalanine in the way. And in this case, the interesting story is that we identified one gate, which is sort of correlated with one single dwell time, with one single closed state. So it's not a general change in the open mobility of the channel, but it's really one single dwell time which is affected. The other dwell times are relatively unaffected. So with this, we can move to another gate, because all these viral channels, they have this very similar architecture, very similar pore structure. And in all these channels, we find that at negative voltages, we have this negative slope conductance, which means the unitary conductance decreases in all of these channels at extreme negative voltages. And the reason for this is easily seen in the channel traces. We see the more negative we go, the more flickery the ion channel opening becomes. Here we have nice clear-cut openings, and here at negative voltages, this become very sort of flickering. And when we think a little bit more in theory, we realize that these clear-cut openings, they are recorded by our recording system because they are long-lasting, and even with some filtering, the recording system is able to sort of record the entire opening from a closed state to a full open state. If, however, the gating of the channel is very fast, like in this case here, the recording system, including all the filtering and the acquisition of the data, is slower than the operation of the channel, which results sort of in a cutting off of the real channel amplitude. So the recording system is unable to really measure the true open channel current. It measures something in between. So when we do all point histograms, you see that the full nice opening gives data for the closed state, and it gives clear-cut data for the open state. If you have, however, this flickery gating, we get a nice clear-cut closed state and some skewed data which are spreading the entire area here between the open and the closed state. And when we look at our dwell-time histograms from our channels, we can basically identify 3 different types of gating. We can identify a slow gating, which is fully sort of seen in defined closed states like here. We see a second slow gate, which is sort of an intermediate time. We see these medium-long closed times, which are responsible for this flicker gating. And we see some very fast gating, which you see here in the fully open state. And this is in the range of about 1 to 10 microseconds. This medium fast gating, which will be the topic of the next slides, is in the range of about 50 to 150 microseconds. So we can identify 3 distinct gates, which are different in their velocity. And from this, we can derive a simple Markov model with 1 open state and 3 different closed states: a medium long, a fast, and a slow closed state. So what can we do with this now? We can extract information from this very fast gating data. And this is the work mostly of Peter Hansen, who has been pursuing this analysis for more than 30 years in Intra Schröder. What we do is we record the data and we use the Markov model. With the data, we generate these all-point histograms, and we use this Markov model to simulate the same distribution. So we consider, in this case, all the filtering, all the noise, and the rate constants of this model to regenerate or to simulate as good as possible the experimental data. So the black line is what you measure, and the red line is what is simulated. So basically, we reconstruct, we refold from this noisy data the real open state of the channel and all the rate constants which are underlying this activity. And with this analysis, we measure this unitary conductance with this negative slope, and we can reconstruct the amplitude of the channel as it is really opening. And you see this is a nice linear ohmic-like conductance. And we can even test this together with Andreas Hartl and Ken Shepard from Columbia. We used their high bandwidth amplifier. And you see with a 10 kilohertz sampling, these are the red data here. They come very close to what we predict from our analysis method as the real open channel amplitude of the channel. But they were also then filtering the channel or their data, they measured data with 1 kilohertz, which corresponds to our sampling of the data, and they regenerate the IV curve that we measure. So what we measure and what we predict from our analysis is very well reproduced by high bandwidth measurements, so we can be sure that our analysis is a good sort of approach for extracting information from this very fast gating here. So now we not only get the information of the full open amplitude of the channel, we also get all the rate constants from this Markov model, including the ones which are responsible for this flickery type gating. And when we analyze this and we plot them as a function of voltage, we see that this is a very strong voltage-dependent process. Here you have nearly 2 orders of magnitude in rate constants change for this 100 or 200 millivolt here in voltage. So the interesting thing is, so we have a voltage-dependent channel which has no voltage sensor. So there is some inherent voltage dependency in a potassium channel. And this is now the question, where is this gate, this voltage-dependent gate, where is it located? So the best guess, and I will convince you that this is a good guess, is that this voltage-dependent gate is the filter of the channel. The first argument is that this flickery gating of the channel is sensitive to potassium. The higher the potassium concentration here between 100 and 1.5 molar is, that this fast gating or the resolution of the full opening is moved to further negative voltages, which means that the potassium is compensating the lack of resolution of this fast gating. So this means that the ion which is transported is also influencing this gate, and this argues for a location of this gate in the selectivity filter. And this is further supported by some mutations that we did, and we found more by serendipity. When we mutate this serine here, which is behind the selectivity filter, and actually it is in its position equivalent to this E71 mutation in KCSA, which interferes with the selectivity filter, in this case not like in the KCSA via a salt bridge, but by hydrogen bond networks, we can influence the stability of the selectivity filter. And you see that this mutation here completely removes this flicker gating. Now we are able to move and see the entire opening of the channel, and the rate constants for closing here decreases by 1 order of magnitude. So all this argues for a position of this gate in the selectivity filter. And now we can do experiments. So we can do experiments over a wide range of potassium to see the influence of potassium on this gate. So we have been doing experiments in symmetrical solution from 50 millimolar to 1.5 molar. And you see here the curves, a lot of work. And the first information that we get from this is that at low concentrations, these IV curves are very linear, which means that the voltage-dependent step is not rate limiting. At very high concentrations, we see some exponential here form of the curve, which means that only at very high concentrations and at very negative potentials, this voltage-dependent step becomes rate limiting. We have also been doing experiments in which we kept the external concentration stable, 100 millimolar, and varied the concentration on the internal side, or in which we kept the internal concentration stable, and we varied the concentration of potassium chloride on the external side between 100 millimolar and 1.5 molar. And what we find is that the the channel is rather insensitive to the changes on the internal side. All these changes which are affecting the voltage dependency of this channel, they are related to exchanges of potassium on the external side. So what we see is the channel is asymmetric and or has an asymmetry, and it has an external side or recognition side or binding side for potassium. And now, if we have good data, we can also go and do the next step, and we call this model-based IV relation analysis. Means that we take this data and try to extract from them the structural information. How does a potassium ion go through the filter of a potassium? So we have this 5-state model here, which has been proposed by Benoit Roux in 2005 for the KCSA channel. So we can do the kinetic model of this channel, of this model here, and compare it to our measured data. So this is, for me, a very impressive figure, because in this case, we have been fitting 36 data sets for different concentrations, different conditions, external versus internal symmetric solutions, the IV curve and the rate constants all together with one model. And you see that the fits are really surprisingly good. So we find all the sort of the nuances of the data, the steepness of the relationship of the rate constants, the non-linearity of the IV curves, the sideness is all reflected very well between data and simulations. So with this, we can confirm this 5-state model as a good model for explaining the experimental observed data on potassium channel measurements in this channel. But if we have a model analysis, we can also analyze alternative models. So we have been reducing this 5-state model to a 4-state model and removed this S0 binding site here. And or as an alternative, we use the hard knock-on model, which has been proposed by the Crowd group, here again as a 5-state model, including this S0 site. And the conclusion is that all of these alternative models, they were inferior to the soft knock-on 5-state model of Benoit Roux. And here you see that here the hard knock-on model is not as good as making a good prediction for the, or is not well fitting the rate constants. They are much too fast. So there are good reasons to believe that at least in our case, for our channel, the 5-state model with an external binding site S0, which is actually the sensor for the potassium concentration, is the best model for explaining this large set of data. And we think that this is a good description for what is happening in the potassium channel, and that the fast gating is nothing else but a collapse of the pore in the case that the supply of potassium through this S0 site is not fast enough, and the pore runs free of potassium. So now we can even ask the additional question: how much are these serines here contributing to this gating effect? And we can make concatenamers in which we exchange the single serines or double or all the 4 serines through to a threonine, and we can see how much this individual or combination of mutations are contributing in a concatenamer to this pore gating. And the results are very nice because they're very simple, which means the more mutations we introduce, the lower the rate constant. And this is quasi a sort of monomer mathematics. So each of the mutations is contributing about the same amount of slowing down of this gating, so that we come to the conclusion that each of these serine to threonine mutations is lowering the barrier for in this hopping model. So with this, I'm coming to my last slide. We have been using very, very simple proteins, ion channels. We combined them with high resolution measurements and data analysis. And with this endeavor, we could identify single gating mechanisms and identify the physics and the chemistry behind this sort of mechanisms or conformational arrangements in the protein, which are responsible for single gating events in these simple proteins. And with this, I close my talk, and I'm waiting for your questions.
Okay, thank you very much, Gerhard. Very nice talk. We have few questions. Let me scroll down the chat. So, okay, the first question is relates to the I think the first part of your talk is from Gianfranco Bochinfuso. He asks, how much common is this hinge when a serine is present in a helix in the PDB? How much common?
Oh, this is, I forgot the number, but in this paper by Leonardo Pardo, he was going through published PDBs and of membrane proteins, and I think in 24% of the structures he found serines or threonines in which he sort of found the potential for this kink forming activity, including ion channels.
Okay, okay. Sorry?
No, but the number is sort of can be extracted from the paper. I don't know by heart anyway.
Okay. Gianfranco also was asking, probably we have to observe the serine each time that the hydroxyl in the serine cannot do an H bond with other residues of solvent, isn't it?
You know, the thing is, this serine conformation is only valid if it's in the helix, and if it's in this gauche position. It's not any serine is able to do this helical interhelical bond. So it's not that every serine or threonine is a candidate for this. And it also has to have the space around. So there must be the possibility that the alpha helix can also, if it's tight, if the environment is not allowing any kinking, it will not happen.
Okay. So next question is from Luigi Catazzano. Is about the second part of your talk. How do you define a knock-on model? Okay.
Well, the soft knock-on model is with the water in between the ions, and the hard knock-on is one ion touching the or close to the other. So if each of the sides in the filter is occupied by an ion, this is the idea of the hard knock-on. And the soft knock-on is if there's a water in between the, and this in our calculations depends or makes a different occupation probability of the different sites.
Okay, so there are still a lot of other questions, but we have to switch to the next speaker. So I invite the others to write in the private chat to Gerhard to ask their questions.